Good afternoon, everyone. My name is Sabrina Elkassas, and I am excited to talk to you today about my research studying subseafloor chemolithoautorophs as models for understanding the potential for life on ocean worlds. There are several different ocean worlds in our solar system, some of which are shown here. You have probably heard of Enceladus and Europa, which are both very heavily studied ocean worlds. Here is a profile of Enceladus, one of the main ocean worlds that I study, which I'll use as an example. Ocean worlds in our solar system have thick and icy exteriors with liquid water below. This means that sunlight cannot penetrate to the depths of the liquid water, so survival of life here would be dependent entirely on chemical energy, just like at hydrothermal vent systems here on Earth. Going a bit deeper into detail, Enceladus has a chondritic core, which means that it's undifferentiated. It's covered in 2 to 60 kilometers of ice. There are no plate tectonics, but the water beneath the ice is heated by tidal deformation. This tidal deformation creates circulation of this water, or flow, which is key for rock-water interactions that create energy for life. This water and accompanying compounds are released as plumes in the south polar region of Enceladus, where the Cassini probe detected that the plume contains chemicals that microbes here on Earth use for their metabolisms at hydrothermal vents. So a key question that we were investigating is, how is axial seamount, our hydrothermal vent study site, an analog environment to other ocean worlds? First, deep-sea hydrothermal vent systems are where heated water flows through crustal rocks. This process alters the composition of both the fluid and the rocks it flows through, creating a chemical disequilibrium that provides impetus for chemical reactions to occur. Life at these depths, without sunlight, like ocean worlds, can capitalize on these reactions to power their metabolisms through chemosynthesis. In our lab, we specifically focus on sampling diffusely flowing vents. They are a mix of vent fluids and seawater at less than 120 degrees Celsius, so much lower in temperature than the black smoker you can see in the picture there. They are the largest source of new carbon in the vent ecosystem, containing 5 to 10 times more cells than deep water, and holding microbial assemblages at the crustal interface. Thus, by sampling the water from these vents, we also get a window into life just underneath the subsea floor. Our study site, axial seamount, is a hydrothermal seamount situated on top of the Juan de Fuca Ridge spreading axis. The 3 diffuse vent sites of interest within axial were Anemone, marker 33, and marker 113. Each of these sites was sampled each year from 2013 to 2015. This gave us the opportunity to conduct not only spatial analyses, but also temporal ones. From the different chemical measurements of each of these vent sites in 2013, we can see that the vents are similar in both temperature and pH. The largest difference is seen with the hydrogen concentration, which is much higher at Anemone and below the limit of detection at marker 113. We'll see later that this is because there are a large number of methanogens at marker 113, which draws down the hydrogen level and increases the methane, which you can also see in the table there. The oxygen at marker 113 is half the concentration of the other vents and the lowest measured across all vents sampled. I've highlighted hydrogen and methane again as a reminder that both of these compounds were detected by the Cassini probe in the Enceladus plume. So A lot of studies now focus on studying methanogens, which use hydrogen and carbon dioxide and produce methane. In this cross-year study, a combination of approaches was used to determine the active subseafloor autotrophs over space, time, and geochemical gradients, and I am currently still analyzing that data. Metagenomics was used to understand the potential functions and taxonomy in the sampled populations. Metatranscriptomics was used to identify active functions within the sampled population. To further tease out the autotrophic signal, RNA Stable Isotope Probing was used. In this experimental technique, isotopic incubations label only active autotrophs in the sample. Then further metatranscriptomic sequencing allows us to determine the identity of those members of the microbial population. The goal is to construct full metabolic and functional profiles at all sites. In the next few slides, I'll go into detail about how RNA stable isotope probing works and why it is such a powerful tool. RNA stable isotope probing starts with the incubation of vent fluid, which you can see in the upper right, along with a labeled substrate. In this case, we use 12 or 13C labeled bicarbonate. The controls and experiment are labeled at different temperatures, and we use 3 different incubation temperatures, 30, 55, and 80. Then the RNA is extracted, quantified, and fractionated using an ultracentrifuge. And in the ultracentrifuge tube, after spinning it for 3 days, the heavier fractions are at the bottom, which are the microbial populations that took up the 13C label, and the lighter ones are at the top, which are not the active autotrophs. 12 to 16 fractions are collected, and then the RNA is precipitated and quantified. Graphs like that shown on the bottom left are made to understand which fractions to take through metatranscriptome sequencing, which we'll talk about on the next slide. From the density fractionation and subsequent RNA precipitation, we create these graphs. The green line shows the control with the 12C labeled bicarbonate, and 12L means that it's light in the 12C labeled bicarbonate. The orange line shows the result of the 13C incubation, and the 13H means that it's heavy in the 13C label. And as you can see, the peak buoyant density is shifted as compared to the control in this orange line from 1.78 to around 1.81. So we then sequence the metatranscriptome of the 13C heavy fraction to identify the active autotrophs, which are those which took up the labeled 13C bicarbonate. So now I created these graphs to compare results over space, time, and different geochemical gradients. So this one is over space across all 3 vent sites at a single temperature of 80 degrees C during the year 2013. Marker 113, which is in the middle, is heavily dominated by Methanococcus, which is a methanogen, and which is why earlier when I showed the drawdown of H2 and the increase in methane, it's due to this domination of this methanogen. Marker 33 has few to none Methanococcus. Likewise, Thermovibrio is present at marker 33 and anemone, but does not appear at all in marker 113. So next I compared RNA SIP results again over space. However, this time it was for 80C, so the same temperature as before, during 2014. This allows us to compare across both space and time because we can compare it to the 2013 results as well. First, all of the Methanococcus we saw at marker 113 is gone, again, it's in the middle bar. Now, all 3 vent sites have a similar composition of Desulfurobacterium, Thermovibrio, and an unclassified member of the family Desulfurobacteriaceae. This shows that hydrothermal vents can vary quite a lot in their microbial community composition in just 1 year. This should be kept in mind when we capture snapshots of the geochemical composition of hydrothermal vents on other ocean worlds. Just because we measure something once doesn't mean it can't change drastically in a very short amount of time. Next, I looked across time. At one vent site, marker 113, and at one temperature of 55 degrees Celsius. Unlike at 80 degrees Celsius where we saw Methanocaldococcus one year and did not see it in the next, here the microbial community composition remains fairly constant over the 2 years, with a slight decrease in diversity in 2014 as compared to 2013. Finally, I looked across temperature at Anemone vent site in 2014. The rightmost bar represents the entire metatranscriptome at this site, which you can see contains many members. However, at each temperature, we see not only a distillation of the members in this metatranscriptome, but also that just a few families dominate at each temperature. I did not show it by genus this time since the metatranscriptome becomes even harder to distinguish. At 30 and 55 degrees C, members of the family Nataliaceae dominate, whereas they are absent at 80 C. At 80 C, we see members of the Decilpherobacteriaceae and Aquificaceae families. The key takeaway from this talk is that both axial and Enceladus or other ocean worlds have similar environments: lack of light, rock-water interactions at hydrothermal vents, chemical energy for life, and similar temperatures. With this study, we were able to create profiles of active autotrophs at variable chemical regimes, which lends us predictive power of what autotrophs would exist in a given environmental regime on other ocean worlds. This will help inform efforts to understand and detect life on other ocean worlds. Thank you.